Welcome, everyone. I look forward to your participation today. So um, we will get started in just a few minutes. And if you can put in the chat, let us know where you're dialing in from today. That would be great. I want to see how far of a reach we have today. We've got Camellia in San Diego. Welcome. We've got Michael in New York. And we have Dave in Chicago, but he's not dialing in. Okay. Well, welcome everyone. Thank you for attending our first exhibitor webinar series. We are graciously sponsored today by our partner, SCIEX. So today's presentation is on the advancement of capillary gel electrophoresis through the use of native fluorescence detection. Our speaker today, I'll get to that in just a moment. We just want to remind you that the views and opinions expressed during this webinar are those of the presenters and do not necessarily reflect the official policy position and views of CASSS. And the webinar is intended for informational and educational purposes only and should not be construed as legal, medical, or professional advice. And our presenter today is Jessica Taylor, Senior Research Associate at Teva Pharmaceuticals. She specializes in analytical chemistry there. And in the past 4 years in the biopharmaceutical industry, Jessica has focused on growing her expertise in separation science, particularly in the CE field, including capillary gel electrophoresis, capillary zone electrophoresis, capillary isoelectric focusing, as well as imaged capillary isoelectric focusing. Jessica continuously works to optimize and refine methods where possible to continue to drive innovation in the CE space. So welcome, Jessica. Thank you so much for being here. I want to thank Roxana McCloskey as well at SCIEX for being here. And before we get started with Jessica's presentation, we just want to take a quick pulse of the where you feel you have some questions here. Oops, sorry, I'm trying to get to this slide. I'm going to share my screen again. And it's a little poll, so you can scan your QR code there, or you can join at menti, menti.com, and use the code up here at the top of the screen. What one word describes your biggest challenge or interest in CE? We'll give you folks a minute or so to start adding your words, and we should see them pop up here. Okay, I'm not sure why they're not showing up. Oops. Okay, well, this was did not go as planned here. So for some reason, it's not showing the word cloud. I apologize. I will figure this out and we'll bring it back a little later in the presentation. So I'm going to go ahead and stop sharing and allow Jessica to start her presentation.
Right. Just bear with me a second while I get my screen shared. All right. All right. Can everyone see the presentation?
Yes, we can.
Okay, awesome. So thank you, Anne, for the introduction. As she said, my name is Jessica Taylor, and today I'm here to discuss the advancement of capillary gel electrophoresis through the use of native fluorescence detection. So this here is just our legal disclaimer slide. And then I just want to start by giving a brief agenda of the things that I'll be covering today. So I'm just going to start with a brief introduction to my team at Teva, as well as the native fluorescence technology. Then we'll jump right into some data, looking at some peak-to-peak comparisons between samples run using the UV detector as well as the native fluorescence detector. Then we'll look at some stress and enzyme digested samples, again, run using both detectors. And then we're going to look more into kind of interrogating the native fluorescence detector a little bit more by looking at some things like loading linearity and LOQ. And then we're going to look at some potential applications for the native fluorescence detector with low concentration samples before finally wrapping up with a brief summary and conclusions. Oops. Okay, so I'm part of the analytical development team at Teva, which is part of the larger biologics CMC organization located in Westchester, Pennsylvania. Our team supports non-GMP release and stability studies. We then qualify our methods and transfer them to our QC group for formal release and stability testing. So non-reduced and reduced CGE sample analyses are routinely performed in our group to support our process development and formulation development teams. So 2 years ago, our lab brought the 8 capillary SCIEX Biophase 8800 system online to support high throughput studies. The Biophase has given us a significant improvement in throughput for really large early phase studies, but we have a bit of a hang up still on the quick turnaround of data due to the data processing side of things, just because of the amount of manual integration that's typically required for processing CGE data. So native fluorescence detection has the potential to greatly improve the integration of CGE data. So NFD works by employing a molecule's native fluorescence capabilities from its aromatic amino acids, which are found on the bottom right corner of the screen here. So NFD eliminates any baseline noise or interference that we might see in our typical UV spectra. And it also doesn't require any additional labeling like that which you would use for laser-induced fluorescence methods. So SCIEX approached my team at Teva to perform a pre-commercial evaluation of the SCIEX Biophase equipped with the native fluorescence detector to examine any analytical distinctions between the two detectors across a range of sample type under practical laboratory conditions. So the bottom left-hand corner of the screen shows a schematic of the Biophase, now including that new native fluorescence detector. And because of the way the instrument is set up, you can only use one detector at a time. So you can use both within one sample set. But if you wanted to collect native fluorescence data as well as UV data for a single sample, you'd have to do one injection and collect data using the UV detector and then perform another injection to collect the data with the native fluorescence detector. So to begin our evaluation, we took 14 of our in-house Teva molecules along with NISMAB and prepared them under non-reduced and reduced conditions. And then we ran them using both the native fluorescence detector as well as the UV detector. So starting off here with a non-reduced example, I would like to point out that all of the electropherograms that I'm going to show throughout this presentation have been normalized. The native fluorescence detector has a much higher signal intensity than the UV detector, So in order to be able to overlay them in a meaningful way, I did normalize all my electropherograms to the highest peak in that egram. So for this particular example, if you look at the zoomed electropherogram present in the upper right corner of the screen, you can see the UV data in the black trace and the native fluorescence data shown in the pink trace. Now for this sample, you can see that all the minor peaks detected using the UV detector are also visible using the native fluorescence detector. Now, if we look at the numeric data for this same sample, you can see in the left side of the table that the reported results for this sample are comparable using both detectors. One of the major advantages for the native fluorescence, particularly in non-reduced samples, is the lack of baseline interference in this high molecular weight region. So for this particular sample, the high molecular weight peak is this peak 6. And if you look at the individual data present in the right side of the table, you can see that the variance for the detection of this high molecular weight peak is significantly decreased using the native fluorescence detector. Now here is an example of one of our reduced molecules. And you can see in the zoomed view in the upper right that once again, all of the peaks present in the UV trace are also detectable using the native fluorescence detection. And if we look at the numeric results for this sample as well, you can see that the reported results present on the left side of the table are comparable between both the detectors as well as the individual peak data between both detection mechanisms. Now, if we look here at another reduced example, you can see in the zoom view in the upper right that once again, any peak that we see in the UV trace is also detectable using the native fluorescence detector. But for this example, we did see a higher heavy chain to light chain ratio using the native fluorescence detector compared to the UV detector. So what that means when we look at the numeric data for that sample, if we look at our individual light chain and heavy chain peaks, which I have boxed here in the red, you can see we see a roughly 6% increase in our percent heavy chain and a corresponding 6% decrease in our percent light chain. However, if you look at our reported results on the left side of the table, you can see that they do remain comparable between both detection mechanisms. So the reason we see this difference here is due to the amount of aromatic amino acids present in the molecule. So this molecule in particular has 10 tryptophans in its heavy chain, but only 2 tryptophans present in its light chain. Now, the molecule that we looked at a couple slides ago, where we didn't see this heavy chain to light chain ratio discrepancy, had 9 tryptophans in its heavy chain and 4 in its light chain, which led to a more comparable heavy chain to light chain ratio between the two detectors. So after looking at these 14 molecules and seeing comparable data between the two detectors, we then wanted to look at some stress material to see if when we generate new fragment peaks, would we still be able to see all of the peaks that we see in UV using the native fluorescence detector? So here we have an example of a reduced stress material. This material was stressed at 40 C for 8 weeks. And if we look at the zoomed electropherogram in the upper right, you can see that all of the peaks that appeared under these stress conditions in the UV were also visible using the native fluorescence detector. Now, I did want to point out here that if we saw that discrepancy in the heavy chain to light chain ratio in the unstressed material, you would see that same kind of increase in the heavy chain to light chain ratio in the native fluorescence in the stress material as well. But if you did see this difference in the heavy chain to light chain ratio, once again, your reported results would still remain consistent between both detectors. So next, I want to look at generating some fragments of additional sizes. So here I took a standard sample and I digested it using the IDS enzyme. This cleaves the MAB at the hinge region and creates the FC and Fab2 fragments. So this sample is a digested sample prepared under non-reduced conditions. And if we look at the zoomed electrophoregram in the upper right corner of the screen, you can see that once again, all the peaks you see under the UV conditions, you can also see using the native fluorescence detector. And actually, in this case, with the native fluorescence detector, we could see an additional fragment peak at about 30 minutes here using the native fluorescence detector that we were unable to detect using the UV detector because of the baseline noise in that region. And if we look at the numeric data for this particular sample, we saw mostly comparable results between the two detectors, except for when we got to the larger peaks. So those I have boxed here in the red. And you can see there's a little bit of some discrepancies between the percent TCA data for these larger peaks, kind of similar to what we saw in the reduced CGE results when we saw that increased heavy chain to light chain ratio in some molecules. And similar to that, when we add our major peaks together, our reported results shown over here on the left do remain comparable between the two detectors. So this is that same sample now prepared under reduced conditions. And if we look at the zoomed electrophoregram in the upper right, you can once again see that the peaks present in the UV were also detectable using the native fluorescence detector. And if we look at the numeric data for this sample, we did see comparable results similar to what we saw in non-reduced until we looked at kind of the larger peaks where we saw some discrepancies again. But once again, when we summed these major peaks together, we did see comparable reported results between the two detectors. So at this point in our evaluation, we were fairly confident that any peak that we would see under UV conditions, we would also see using the native fluorescence detector. So now we kind of wanted to move on to looking at the native fluorescence detection technology as a whole and any potential applications that would have to our assays. So we started by looking at loading linearity. So I took one of our molecules and I prepared it at 7 final protein concentrations under both non-reduced and reduced conditions. Each concentration was then injected 3 times and detected using both the UV and the native fluorescence detector. I then averaged the 3 injections and used that to determine the linearity of each detector. So the results are shown in the graphs here on the left. The top graph shows the results for the UV detector, and the bottom graph shows the results for the native fluorescence detector. In both graphs, the blue line represents the non-reduced assay, while the red line represents the reduced assay. So looking at the UV data first, you can see that we had good linearity for both the non-reduced and reduced assays with R-squared values of about 0.98 for both of them. Now looking at the same data for the native fluorescence detector, you can see that we had slightly lower linearity with R-squared values of about 0.94 for each. We're not sure exactly at this point what caused the difference between the two detectors. Thus far, we've only tested loading linearity this one time with this one molecule. So there's potential to see some different results if we were to repeat this testing or test this again with one of our other molecules. So we wanted to take the data from our loading linearity study and use it to investigate any potential differences in LOQ that might exist between the UV and the native fluorescence detector. So we did that by taking 3 minor peaks based on where they migrated in the UV trace. So if you look at the electrophoregram at the bottom of your screen, we first selected peak 1, box here in the green, because it migrated in an area of relatively low baseline interference in our UV trace. We also looked at peak 4, which migrated in an area of, I would say, moderate baseline interference in our UV trace. And then finally, we selected the high molecular weight peak 9, because it migrated in an area of notoriously high baseline interference in our UV trace. So then for each of these peaks that we selected, we calculated precision and accuracy data throughout our tested range of 0.75 to 2.25 mg per mil. This represented 50 to 150% of our target concentration of 1.5 mg per mil. We then calculated our LOQ by taking the average area of our minor peak at the lowest level of the range that showed passing precision and accuracy data and dividing it by the average total area at the target level of our range. So now we're going to look at those results peak by peak. So at the top of the screen in the black, you'll see the results for the UV data. And at the bottom of the screen in the pink, you'll see the results for the native fluorescence. So starting off here with peak 1, we got passing precision and accuracy data throughout our tested range of 50 to 150% of our target load, which gave us a calculated LOQ of 0.03% for both the UV and the native fluorescence detector. I would like to point out that with the native fluorescence detector, we did see tighter precision data, particularly at the lower ends of our range. Now moving on to peak 4, we once again saw passing precision and accuracy data throughout our tested range of 50 to 150% using both the UV and the native fluorescence detector. So this gave us a calculated LOQ of about 0.1% using the UV detector and a slightly lower calculated LOQ of 0.08% using the native fluorescence detector. And once again, here we did see tighter precision at the low ends of our range using the native fluorescence detector. And finally, moving on to that high molecular weight peak, peak 9, using the UV trace, we saw failing precision and or accuracy data at the low ends of our range. So for this peak, our range was 133 to 150% of our target load. And this gave us a calculated LOQ of 0.11%. Now, looking at the data for this peak for the native fluorescence detector, we had passing precision and accuracy throughout our entire range of 50 to 150%. And this gave us a calculated LOQ of 0.07%. So from this data set, we concluded that the native fluorescence detector may not necessarily give you a significantly lower LOQ than that you would get in the UV. But you do tend to see tighter precision data, particularly at the low ends of your range, due to the consistency of integration from the lack of baseline interference. And for any peak that you have that migrates in an area of particularly high baseline interference, you will likely get improved precision and accuracy data throughout your range. So we also want to look at the potential for native fluorescence detection with low concentration samples. So low concentration samples are fairly common due to highly potent drugs that need to be delivered at lower doses, requiring lower DP concentrations. Some molecules also have downstream processing occurring at low concentration and in non-formulated matrices. We also have clinical in use studies which mock protein delivery in intravenous delivery systems at low concentrations and in different matrices, such as dextrose and saline. So with our typical UV methods, my approach to testing these low concentration samples might include doing things like modifying our sample injection to either extend the injection time to allow the sample more time to enter the capillary, or switching from the traditional electrokinetic injection to a pressure injection. I may also modify my sample preparation to increase the final protein concentration in the prepared sample. And if I'm worried about matrix interference, I might do a buffer exchange to put the sample in a more CGE-friendly matrix. And if none of these modifications are desirable, or if I'm working with a limited amount of material, I may also utilize an orthogonal chip-based CGE method that operates at a lower protein concentration. So to test the native fluorescence detector's abilities kind of in this space, I took one of our molecules and prepared it at our standard protein concentration of 1.5 mg per mil. And then I also took that same sample and prepared it at a lower concentration of 0.15 mg per mil using either water or 1x PBS as the diluent to kind of test the interference of a salt matrix as well. So this slide here shows the results for the UV detector for the sample set. If you looked in the zoom view in the upper right corner of the screen, you can see the standard sample in the black trace and the low concentration samples in the blue trace. The dark blue trace shows the low concentration sample prepared using water as the diluent, and the light blue shows that same low concentration sample prepared with 1x PBS as the diluent. So as you can see in the zoom view, in the low concentration samples, we can only detect our major HHL and IgG peaks. Other minor peaks are not visible. So in order to test samples at these concentrations using a UV detector, we would have to do some type of modification to the sample preparation and or the injection to boost signal so we could properly analyze the sample. Now this slide here shows the results for those same samples now detected using the native fluorescence detector. So the pink shows our standard sample at 1.5 mg per mil, and then the purple and the orange show the low concentration samples. So as you can see in the zoom view here, you can still see all of the minor peaks in both our low concentration samples. So to test samples at these concentrations using a native fluorescence detector, you might want to use the modified sample preparation or injection methods for ease of analysis, but you don't necessarily have to as you would for the UV detector. So now just to kind of wrap up after performing this evaluation and our thoughts regarding the native fluorescence detector, particularly in its applications in the QC space. So the current practices for GMP discourage the use of manual integrations. So the way that most QC labs work involves tailoring a processing method to each sample set to properly integrate samples and account for baseline changes without needing manual integration. So native fluorescence detection could potentially significantly improve this workflow because of the lack of baseline interference. So this would allow automatic integration to be possible without having to modify your processing method every time. This would lead to notable time savings in data processing for both the analysts and the reviewers. And because of the lack of baseline interference, you would also see improved precision due to integration consistency and provide confidence for the analyst when identifying any new peaks that may appear in an electrophoregram. However, to best fit the current GMP workflows, the native fluorescence detector would need to be paired to a single capillary systems. Most GMP labs just simply don't have the need for the high throughput of an 8-capillary system. So just to wrap up with our final thoughts from our evaluation, thus far in our hands, all the peaks that we've seen present in a UV trace have also been detectable using the native fluorescence detector. However, assessments may need to be done on a molecule by molecule basis before using the native fluorescence detector for routine use. We've also seen comparable reported results between both the UV and the native fluorescence detection. But some molecules do show an increase in the heavy chain to light chain ratio when using the native fluorescence detection. But again, even if you do see this, your reported results do remain comparable between the two detectors. You could potentially improve the range of your assays by using the native fluorescence detector. So you could potentially extend your range to include lower concentrations if we're using the native fluorescence detector. And because of the increased signal intensity with the native fluorescence detector, we also help to decrease the complications of interference from things near matrices like salts. And as we move forward with using the native fluorescence detector, some potential rooms for improvements that we saw was currently in the biophase software, you cannot normalize the UV and NFD signals. So as I said earlier in the presentation, the native fluorescence detector has a much higher signal intensity. So to be able to overlay them, you have to normalize the traces so that you can see both in one overlay. So right now, what you have to do is first export your data and then import it into a third-party software such as Empower, where you can then normalize them and overlay them. We would also love to see the native fluorescence detector paired with a single capillary instrument. As I said previously, this would allow for applications in the GMP space, as well as kind of help with implication in our day-to-day workflows where we don't always need the high throughput capabilities of an capillary system. And finally, we would love to maximize the injections we get per sequence by having simultaneous collection of the UV and the native fluorescence data. Now, in this presentation, I've focused on the native fluorescence detector's capabilities and advantages in the CGE assays. But you could also apply a lot of these advantages to charge variant analysis as well. That you can run on the biophase. So that would include CIEF as well as some CZE. So I would just like to thank these people from Teva as well as SCIEX for their support throughout this project. And thank you everyone here today for joining and for your time. And I would be happy to take any questions that you guys might have. I can stop sharing my screen.
Yeah, so if you have any questions, please go ahead and submit them in the Q&A box. We can also have you answer or ask a question live. If you raise your hand, I can promote you to a panelist and you can have your question answered live. I'll give folks a few minutes to do that.
Yeah.
Thank you, Jessica. That was fantastic.
Thanks, Anne.
All right. Let's see. There we go. All right, we have a question from Joanna Schrofner regarding the difference in HCLC ratio. Do we know what is really the truth value, UV or NFD result? And she also includes background. We see differences in HCLC ratio also with different UV filter age. Do you think this variance we have with UV filter can be omitted by using NFD?
So that's a great question. So the true value, as far as we know, you kind of have a bias in both detectors because, you know, the UV detector is doing it based on the amino acid backbone. So you have a longer amino acid backbone in your heavy chain compared to your light chain, which is much smaller. So in UV, you typically get a ratio of 2:1, where we know for every heavy chain, we have a light chain. So there's already kind of like a bit of a bias there. So you just kind of have, like, I guess, a different bias when you move to the NFD comparing, depending on the amount of aromatic amino acids you have in your molecule. So depending on how that lines up with like the differences in the amino acids chain lengths, you kind of might see like a different bias. So as far as like I'm concerned, like I said, it's just we're always going to have a bit of a bias there. That's kind of different from our true value, which should be a 1:1. But as far as the UV filter age, I do think that the native fluorescence detector could help with any differences you would see there, because it should remain consistent because the number of aromatic amino acids in your heavy chain or light chain, which create the signal in the NFD, are different than what you would see in the UV.
Great, thank you. Seeing if there's any other questions. Some comments. Great presentation. Thank you for an informative presentation. Let's give another minute or two here.
Yeah.
Okay, well, if there's no more questions, we'll go ahead and wrap up. I did have a little challenge with our Mentimeter earlier, so I apologize. But I did put in the chat a couple of the responses that had come through. The baseline noise and reagent consistency were the two of the challenges that folks had mentioned. So thank you for that. So thank you, everyone. We appreciate your time today, and we do have another event coming up in October, so be on the lookout for that. But again, thank you to Jessica and Roxana today for your participation and for supporting the program. We really appreciate it. And this is a fantastic program. Thank you. And this is recorded, so you will receive a recording link when we will be uploading it to our CASSS On Demand site, and you'll have access to it there. Thank you again, everyone.
Thank you, everyone.
Yeah, thank you, everybody. It was great sharing some data with you all.
Have a great day.